So we're looking at the section titled Regulating the Cell Cycle, okay, in your note packet. Now, I've got some questions for you, a little scenario, if you will. How do your cells know when to stop reproducing? Because we're studying cellular reproduction. So suppose you had a paper cut on your finger. Although the cut may have bled and stung a little, after a few days it will have disappeared and your finger would be as good as new, right? After a while you don't even notice you had the cut. But those cells were damaged. So how do you think the body repairs an injury such as a cut on a finger? Well, it obviously grows new cells to fill in the gap, right? How long do you suppose that process actually takes? Well, if it's a paper cut, probably a day or two, maybe, right? It doesn't take that long, and then before you know it, it's gone. But if it's a larger cut, takes significantly longer, right? The bigger the gap, the longer it takes. So what do you think causes the cells to stop the repair process? How do they know when to stop? Why don't they just keep growing and growing and growing and reproducing and reproducing? That's what this section is all about. So imagine we took some skin cells and we grew them on a petri dish, right? Those cells would continue to grow and grow and grow until they filled the whole petri dish. Now let's say I took a razor blade and went and swiped across there and got rid of those, almost imitating the paper cut. Well, then we've got the initiation of those cells near that region where the damage occurred begin to go through cellular reproduction. They go through the cell cycle, they reproduce, they make more cells until the gap is filled in. And then they stop. Interesting, isn't it? All right. So, looking at your notes now, in living things, cell growth and cell division are carefully controlled. Not all cells move through the cell cycle at the same rate. When we look at skin cells and bone marrow, they grow and divide very rapidly, like every 7 hours. Liver cells grow and divide slowly, only like once per year. Muscle and nerve cells do not divide after initial development. Okay? Once they're formed, they don't reproduce. So exactly what controls the rate of cell division? Well, there's a chemical compound called cyclins. Okay? They are proteins that actually control the rate of cell division inside of your cells. So there are what are called internal regulators. And these are proteins that respond to an event inside of the cell that tells the cell when to stop or when to divide. Some examples would include the DNA replication. When that DNA is reproduced, that triggers the cyclins. That's telling the rest of the cell, hey, I've got an extra set of chromosomes here, or an extra set of DNA, it's time to reproduce. So that is a trigger that comes from inside of the cell. But likewise, and we can mimic that in the lab. So what we can do is we can take a sample of the cytoplasm from a cell in mitosis, insert it into another cell that is not already starting to reproduce, and as a result, it triggers that second cell to go ahead and enter mitosis right away, whether it's actually ready or not. Now, there's also external regulators, and those external regulators are proteins that respond to an event outside the cell. So examples include growth hormone from the rest of the body, molecules that are on the surface of neighboring cells, because those cells communicate. So if there's no communication, that tells the cell, nothing's there, time to reproduce and fill in the gap. Now sometimes things can go wrong. And when that happens, one example is cancer. Now I can imagine that most of you have a family member or a friend or friend of the family or something that has had cancer, so you know what that experience is like. It's a disease in which some cells lose the ability to control their cell division. As a result, the cells divide uncontrollably. They never stop. They just keep reproducing and reproducing and reproducing. Now, a tumor is what we would consider an abnormal mass of those cells that have lost the ability to control their cell division. A benign tumor is when the cells of the tumor stay connected to one another and they are not considered cancerous. okay? A malignant tumor is when cells of that tumor literally break off and travel to other parts of the body and then grow elsewhere. That's when we would clinically identify somebody as having cancer, unfortunately. So here's an image of some cancerous cells. You can see that some of them are really abnormal in terms of their size because they're significantly smaller, but they've got 2 nuclei in them. Look at this one back here is already splitting almost into 4. Okay, so that makes it difficult. Now, the reason someone might develop cancer could be caused by 2 different things. One might be a carcinogen, and that's anything that causes cancer from the environment. So tobacco, exposure to UV light and x-rays, okay? So that's why when you go into the doctor's office, if you have to have x-rays, they put on that lead vest over other parts of your body that don't need to be exposed to the x-ray machine. Now, in some cases, cancer can be hereditary, and so that is going to be caused by what we call an oncogene. That's a gene in your DNA that, if it's expressed, will cause the uncontrolled cell growth. Now, carcinogens can actually trigger those oncogenes to be expressed. So if you already have a family history of cancer, exposing yourself to those carcinogens can actually increase your risk of developing cancer. Oh, I'm not really sure why that just happened to us here, but let me go down because, oh, we didn't get to see that last part. Here you go. This was your last line down at the bottom of the page. Was carcinogens can trigger oncogenes. Not sure if you picked up on that. To be expressed, causing that uncontrolled cell growth or cancer. Sorry about that. That was on there, but I just was spacing out. Sorry about that. Hey, have fun. Have a great day. Bye.